So now that we have our PCR product, we need to make sure it's of the correct size, because sometimes PCR will amplify random amounts of DNA and random parts of DNA, and we're only interested in the 16S rRNA portion of the DNA because that's going to help us identify which bacteria you have. So the first step, of course, is to cast the gel. What I have in this bottle right here is some melted agarose. And melted agarose is going to be the base of our gel, but of course we have to let it solidify in order to run it through a gel. And so the first thing we're going to do, of course, is we're going to open it up and we're going to pour into this apparatus right here. So here is an apparatus with some combs. You're going to ask your instructor exactly what size gel that you're going to cast. And so since we only have a small amount of samples for this run, we're going to choose one of these smaller well trays. And what you want to do is you want to fill this up to where it goes halfway up the distance of the comb. You don't want to go too much higher than that. Okay? And so then we'll let this solidify for 10 minutes, and we will actually load our samples onto the gel. So it looks as though our gel is solidified, and so what we're going to do now is we're going to take the comb out of the gel. And to do this, you have to start wiggling a little bit and pulling ever so gently. Now, once the comb is away, you'll notice that this gel has some wells in it, and that's where our DNA sample is going to go. Now, the gel itself goes into this apparatus called the Gel Electrophoesis apparatus, like so. And what we're going to do here is we're going to load the samples of our PCR products in here, and what's going to happen is this agarose gel will have an electrical current running through it to separate the DNA by size. Now, it turns out all DNA has a negative charge, so the current goes from negative to positive, and it will pull the DNA through this gel by size, because if we think about the physics of it, bigger things, if you're trying to pull them through a substance, will move much more slowly than smaller things that will get towards the bottom of this gel. But if you notice here, we do have wires here and here, and notice those wires are not touching the gel. And so we have to add what we call a liquid buffer that will allow the electrical current to flow from one end to another, that will allow the DNA to be pulled through this gel. And so here we have this liquid buffer, and we're going to go ahead and pour it to fill in both sides of this apparatus and to cover the gel. You just want to add enough to where the gel's covered. Okay? Now, this is where steady hands come into play, because you're going to put your DNA sample into these very, very small wells, and we're going to do so using this pipette. Now, what we're going to use from our PCR sample is 10 microliters of volume. And so I'm going to open up my PCR product. I'm going to depress the plunger here, and then I'm going to suck up that 10 microliters from the tube. Now, this is where steady hands come into place. So if you have had coffee right before lab, you might have shaky hands. So you want to kind of make sure that you go down straight into the well, and make sure you're in the well and press the plunger. You want to do this slowly and methodically, but at the same time pay attention to time, because everybody in your lab class has to load their well. The longer this sits, the more likely it is to come out of the well. So time is of the essence. And you're going to do this for every sample you have. Most of the time it's going to be one sample for a student, but if you had more than one PCR going, you might have multiple samples. So we're going to go ahead and load all the wells. Again, you don't want to puncture the well. You don't want to push all the way down into the well. You just simply want to load your sample. Okay. Now, after I'm done loading all of these samples, you'll notice that I skipped the first well. That first well is something that either your instructor will load or a student that has very steady hands will load, because that standard is going to be some cut-up DNA. Now, why would we add cut-up DNA? Well, that DNA is cut by a known size. And that known is what we're going to compare to our unknown PCR products so that we can verify that our PCR products are the right size. And so that's commonly known as a DNA ladder standard. And so you will see when we take a look at the results here what our PCR lines up to as far as how it compares to the standard. Okay, and so we're going to run this gel for roughly half an hour, and we're going to run it at 100 volts. Now, note that in order for this to run, we have to have a protective covering. Notice that there is going to be electrical charge running through here, so if you decided to touch that liquid with your bare hands, you might become electrocuted. So there's this little tab right here. That tab is going to fit right under there, and then you're ready to look at the settings. So there's a 50 volt and 100 volt. You're going to set it to 100 volt. You're going to look at where the leads are. So remember, we're running from negative to positive, so that's set correctly. And all you do is turn on the on button. As soon as it turned this on, you're going to notice the formation of some tiny little bubbles, and that's going to show you that the current is going. And so we're going to let this run until the dye front, or the color, reaches the end of the gel. So it looks as though the gel is completely done, so we'll turn it off. And now we're going to take it out, and we're going to try to visualize the PCR products on this special machine that's a gel documentation station. So note that this gel looks pretty clear. Now, what we're going to do here is we're going to open up this box, and you'll be able to see on the TV what we're doing. We're simply pushing the gel off the tray onto the actual gel documentation station, and we're going to close it up. Now, right now, all you can see is the gel. However, when we put on this device called the transilluminator, which shines a certain wavelength of light, it's going to be a blue light, we can now visualize our bands. And so remember, here is that ladder. And so this standard right here, it goes 4,000, 1850, and this bright one right here is around 1,000. And the product that we're expecting is going to be around between 1,000 and 1,100. And here are the PCR products. So it looks as though our colony PCR worked for this class, and this is around the size that you see. And this size is going to be used in a calculation to allow us to send this off for sequencing so that we can actually know what the genetic material is for identification of the bacteria. And so this is what you're hoping to see after you've run gel electrophoesis. So now that we have our gel electrophoesis results and you've verified that you have an actual band in the correct spot, now we need to actually send it to sequence. And so in order to send this to sequence, we're going to take our PCR reaction, what we have left of it, and we're going to add some components to it. So I have another master mix here that contains two enzymes called alkaline phosphatase and exonuclease. And so this will end up dephosphorylating and chopping up all the excess DNA except your PCR product, so that all you have in here is your PCR product of interest. So when they decide to sequence it, that's the only sequence that you're going to get out of this tube. And so another relatively simple procedure is you're going to take a certain amount of your master mix. This is going to be anywhere between 1.5 microliters and 2 microliters. Again, you're going to talk to your instructor about the exact amount that you need to use. But again, you simply transfer from one tube, from your master mix tube, the amount of microliters that your instructor tells you, into your PCR tube that you had left over after you ran your gel electrophoesis. And you mix by pipetting gently up and down. Once you have verified that's mixed, your PCR tube is now ready to do another reaction. And so we're going to come back to our PCR machine, our thermocycler, and we're going to do open method. And now it says PCR purification. That protocol will enable us to, again, let the enzyme work at 37 degrees Celsius. That's human body temperature. Lots of enzymes work at human body temperature. Then we're going to deactivate the enzymes because you don't want it to overly chop up too much DNA because it might non-specifically start eating your PCR. So that will get the enzymes deactivated, and then it's going to hold at 4 degrees Celsius until your instructor has a chance to come and grab the actual tubes. And so here we will place the tube in here again. Again, do not run this until the rest of the class has this ready. Simply shut the machine down, hit the next, start run, and it will start automatically for you. And this will take roughly about 41 minutes, and we will continue on with this presentation in 41 minutes. So now that our PCR reaction has been cleaned up using those enzymes, now we're ready to prep this to send it out for sequencing. And so to do this, we're going to have to follow the sequencing facility's rules. And so they say that they need about 10 nanograms of DNA per 100 nucleotides of size. And so your size, if you remember from last time, is about 1,100 nucleotides. So we need approximately 100 nanograms. Now, with most PCRs, you're going to get well over that. And so what we're going to assume is that we only need to transfer 1 microliter of your cleaned up PCR to a new tube, and then fill that up with 9 microliters in order for us to have a total of 10. And so now labeling changes a little bit. So you're going to have a new PCR tube, but now we're going to, in order to keep this straight, because we're going to send all the PCRs from every single class of microbiology in one order, we're going to prepare this handy dandy order form. Okay? So when you're all ready, you're going to come up and fill in one of these rows with your information. So if you have position 1, your new PCR tube is going to have a 1 on the top. So I'm going to go ahead and fill in the number 1 with my very thin Sharpie right on top. Okay? Then it says template. So the template is going to be the initials and numbers that you put on your PCR tube. So mine was AK2. So I'm going to go ahead and write AK2 on this form. The size in base pairs, remember that's going to be whatever size your PCR ended up on your gel. So check with your instructor. Most of you guys should have around 1,100 base pairs. And then the last part is the primer. Now, note during PCR we use both a forward and reverse primer to get a double strand of DNA. When you're doing sequencing, however, you only need 1 primer. So check with your instructor whether it's the forward or reverse primer. And so for the purposes of this, we're going to use the forward primer. Okay? So this tells me exactly for tube number 1 what's in that tube. Okay? And you're going to be coming up here, filling out your information so you know what to put on this tube. But the sequencing facility wants to keep track of all the tubes in one order. And so note then, since this is going to be my order, we're going to write down my initials. So we're going to do AK for Angelo Colaquitas. And then you're going to replicate the number on the top. So I have position 1 over here. Okay, so please note that on the side of the tube, you write your instructor's initials, or AK, whichever we're going to order, and the number on the top. Note, though, that that does not coincide with your initials and your number. Your initials and your numbers go on this form. Okay, so it's very important to keep this straight because the sequencing facility, since this at Yale, is going to want to keep all these tubes in order because they have thousands of tubes that they run at once. So that will keep NWTC's orders in one box, if you will, and then that number will coincide with this list that we have. Okay? And so again, very simply, we're going to take 1 microliter. We'll set our pipettes to 1 microliter of our material. So I take that out, take 1 microliter of my material, okay, and put it into this new tube. And it's going to seem like a very small drop because that's 1/1000th of a mil. And then you're going to take a new pipette tip and put in 9 microliters of water. If it's premixed with your primer, it will be 9. If it's not premixed with your primer, it will be 8.5. So again, check with your instructor with whether or not your water is premixed with your primer or not. But the total volume that should end up in this tube is 10 microliters, right? 1 microliter of your sample, about half a microliter of your primer, and then the rest in water. And so that will be 10 microliters. Once you're done setting this up, you're going to bring this to the front of the room for your instructor to help give this to the lab techs to allow them to send it out for sequencing. And when we get the results, those results will be posted in Blackboard, and then you will blast the sequence. And we'll talk about blasting sequences in lab.